welcome to the last conference on CCC, Ciências Cognitivas e Comportamento. This couple of conferences aimed to bring together people that are giving some part of their lives in understanding data analysis and the importance of behavior and data analysis for understanding different, for different purposes, for different goals. So we had people from organization and environment, from robotics and for other areas. And today we'll have someone that analyzes data with the goal of understanding how our brain works and how it relates with behavior. So it's going from a more applied setting to a more lab setting of using knowledge about data analysis. So I think that most of you know our new colleague, Niccolò Bonacchi. I'm always saying bad with the name, so that's why I looked at him. He was a student here, not from psychology, but was a student here, first in rehabilitation, and then he did a master in psychobiology. And I think that was the way he started to enjoy, to know a little bit about the relation of body and behavior and brain. After that, he experienced some moments in Ictle as a research assistant, very briefly, but then he got the possibility of going to Champalimont. It was opening, it was new at that time, and so he was one of the first ones to help to build that center of research with a very good environment for researchers. I think you finished your PhD around 17, yeah? Yes. And after that, I saw that your publications start to go after the PhD, so you had to be in the lab a lot of time doing research before that. And especially in 22, I was amazed with the number of papers you got. That's because they were always waiting for getting out, but it was there. Everything that is understanding of behavior in a computational neuroscientific way and cognition, even decision making. I think your thesis was in decision making, was with really specific models and trying to map brain areas that were engaged in the decision process, of course with rats, but it's a model of our brain, as I understood. And after that, I think that you start to be really in love with data and what we can do with data, and try not to lose data, not even other losing their data. And so Nico is collaborating with different labs, trying to organize a database for neuroscience and brain activity data in really important details and classifications strengths in order to allow research to develop without people having to go to the lab. They could do that in collaboration. And this is published in Nature Methods, I think. And so, but I think you are going to talk a little bit about that. So I'm not going to talk more about it, just to say that it's a pleasure to have you here and to be presenting your work. Thank you.
Thank you very much, Teresa, for the introduction. And thank you for inviting me. And hello, everyone. And online, if there is someone. Okay, so as Teresa was saying, I'm an old student of the house that recently came back I like the idea of coming back to the places that helped you build your scientific mind and contributing back what you learned outside. And hopefully my tenure here will kind of reflect a little bit of that. So I'm gonna the thing I prepared today to talk about is about the collaboration. I stopped working with them at the beginning of this year. So it's a big collaboration called the International Brain Laboratory. And we are doing things in kind of it's a bunch of systems neuroscience labs that all got together in order to tackle a very specific question. So I realized that the audience was disparate, and I hope, please interrupt me at any moment if you have any questions, any clarifications. It's the first time I'm presenting this in this particular format, so it's very flexible. So I want to start by explaining a little bit what systems neuroscience is. I understand we have people from psychology, people from behavioral sciences, and there's even other types of neuroscience itself. So first of all, the systems neuroscience goal is to understand from structure and function of the brain and how that relates to behavior. So looking at the brain by itself turns out it's interesting in terms of biology, but we really care about how the brain works and how that changes and modulates behavior and vice versa in this kind of loop. So the idea for this collaboration is a very old one that comes from a lot of discussions between the main PIs that are actually our signatories of the grant, but also comes from the lab where I did my PhD, where we started discussing the waste of time that we were all doing, repeating all the same things over and over again, and collectively how we are basically redoing over and over the same thing again and wasting a bunch of time in stuff that should already be kind of standardized and already be state-of-the-art, but reproducible. As you might know, also in psychology, we had this recent crisis in terms of reproducibility, where people actually went back and tried to re-implement experiments from papers and well-known papers that then turned out to be either badly analyzed data or basically didn't work. And if you ever did that, I did that at the beginning of my PhD. I started and I was like, okay, this is how the brain works in this case. Okay, let's do the next step. And you spend 6 months trying to reproduce simple results on an open field with an animal. And it turns out the solution was in a small detail. Furthermore, what we have in systems neuroscience is usually labs are divided in specialties. They either specialize in a particular brain region, or they specialize in a particular technique. And so what happens is that my task for measuring visual or sensory decision-making using visual stimulus is actually not really transferable to auditory decision-making, or to somatosensory decision-making, or to value-based decision-making. So if the behavior is not reproducible, then the neural data, which is also very different, is also very hard to reproduce. So we have electrophysiology measurements, it's a whole world. We have a lot of different techniques for measuring brain data, let's call it that. And with the lack of coordination of all these different efforts, we end up having a very fuzzy view of what the brain actually does and how the brain actually works. So that was kind of the main motivation. Also, there is a bunch of cultural issues in terms of how people usually do collaborations, and how there is not really an incentive to share both data and procedures in the community in general, because of a bunch of different, let's call them academic contexts or academic cultures that differ greatly depending on the institution that you're in. So the idea was for us to actually get together. So there's 22 different labs in different places of the mainly US and Europe. The eastmost is in Lithuania, it's dot over there, and the westmost, I believe, is we have Stanford and UCLA. The goal of the collaboration is to get together, and the idea was, okay, so let's settle on one behavioral task and then record from the whole brain. I don't want a task that is optimized for this thing or that thing, and just look at this small window of the orbitofrontal cortex or of the striatum or whatever your brain region of interest is. Let's have a global view of how all the brain works. Of course, one lab to do this, it would just take 20 years. So the idea of coming together was for that. So let's collect all the data and let's create the first brain-wide kind of neural model of decision-making. And we'll put animals in some tasks, and we'll analyze a lot of data, and we'll solve the brain. That was the idea at the beginning. The way we're doing it, as I was saying, is exactly, we settled on one task, and this one task will be contentious at some point, but we'll talk about it in a second. We're going to record from all of the brain, and this requirement also changed over time, because there are some parts of the brain that are very hard to reach. And we want to go through a whole process of standardizing both procedures and materials. And for that, we needed to develop a common data architecture and ways of actually talking and discussing in real time together with people that are in 8 time zones difference. One of the things that I mostly I mostly push forward in general is working in the open. And I am a very big fan of the open science and open source kind of movement. I don't really think there is a reason for hiding the process of how you arrive to conclusions, because you gain much more just by putting it out there and having people actually contribute from somewhere else saying, oh, I had the same problem and here is a solution for you. And this is actually one of the hardest things to implement, is explaining people that it's okay to show what you're doing, it's okay to show your work as you're doing it, if you declare that this is not a final product, this is a process. Okay, so what did we do? And this project is, we are at the end of the kind of first 5-year cycle and thinking about continuation grants at the moment. But what we did was we decided to do what is here is called value-based decision making with prior. So it's a 2 alternative forced choice task. We're using mice as model animals. They're in a box on top of a wheel, and they have a visual stimulus in front of them. The visual stimulus is a gabber patch that has different level of contrasts, and the stimulus either appears on the right or on the left. And the animal's task is to use the wheel to move the stimulus to the center. So the wheel and the stimulus are coupled. If the animal moves the stimulus to the center, he will get a water reward. If he doesn't move the stimulus to the center and moves it the same amount of distance towards the outside of the screen, he gets an error tone, usually a small white noise, and a timeout. So we ended up with this task, and I have to talk about the prior, basically because we have 22 PIs, each of them very experts in their own particular area, and each of them that have very strong opinions on how you should test experimentally something that they know by experience that it works better this way. The problem is that if you are recording from the whole brain, you cannot optimize the behavior for a particular engagement of one area. So what we ended up doing, what I usually said is, our goal is to keep everyone equally unhappy. So it's not a good task for visual decision making. It's not a good task for people to measure motion. It's not a good task for people that measure value-based stuff and uncertainty stuff. It's just good enough for all of them. And in this way, the prior component of the task is how it came in. And the prior component is basically the animals do these decisions repeatedly, and we introduced a block structure. And randomly what we say is, okay, in this block of trials, the stimulus will come mostly from the right side. So 80% of the time will come from the right side with different levels of contrast. And in another block, which is unsignaled to the animal, it will reverse and go to the left side. So what happens when the animal is presented with a zero contrast stimulus is that he cannot use visual information in order to make a decision. He will have to rely on the knowledge or the impression that he has of where the stimulus are coming from. So it's the integration of the previous trials. How much this is integrated, that's a different question, or how far away in time it's integrated. But they will have to, in order to make good decisions, they will have to integrate okay, I'm in a right block or I'm in a left block. So what happens usually is in the, let's call them 50-50 conditions, you have, this is plotting the amount of choices towards the right as a function of the stimulus going from left to right with different contrasts. So as you can see, the more the stimulus is clear on the right, the more the animal decides to go on the right. We call that a psychometric function, although I know there is a difference in terms, which I was talking to Teresa earlier. And what happens when we actually look at blocks, or what we expect to happen when we actually look at blocks of trial, is that the probability of deciding to go right will be higher when I'm in the right block, and vice versa for the left block. So we have this horizontal shift of the psychometric function. This is what we expected. I'll show you data in a second. One of the main goals for going back to the reproducibility issue is that by design, this collaboration only would work if I can grab data from different labs that are doing exactly the same thing and be able to lump them together. So that was kind of the focus of this first paper, which was the first kind of milestone of the collaboration, which was the standardized and reproducible measurement of decision making in mice. So where we share the whole pipeline and the whole process that we went through, with extensive annexes of conditions of how you house animals, humidity in the air, humidity in the food, and it's just it's a hellhole in terms of, because there's so much that institutions give us for free. You say, oh, I'm in lab X, I'm doing an experiment, I have a vivarium that made a bunch of decisions for you, which will influence your experimental results without you knowing. And so we had to go through the whole process of describing all of these and then actually finding differences to be able to attribute the variance of our results to particular things and then change them so they are all homogeneous. One of the things that was very important for us, and if you ever worked in a lab, you have, or if you haven't had students in the lab, you have better experimentalists and worse experimentalists, better students and worse students. And what we really wanted to avoid is the effect of the golden student, or the golden postdoc, which is, the task is perfect, the results are awesome, but he's the only person who can train an animal, because that person is the only one who knows how to bump on the box at the precise moment so that the animal doesn't get into a weird learning curve and never gets the task. So for that, we made a big effort in actually automating all of our training. So here is an example. We parameterize all our stimuli and increase the level of difficulties that we wanted the animal to go through, and make decisions. Actually, the computer implements decision. You just put an animal in the box, press a button, and our system knows what the performance of the animal and the various indicator was in the previous sessions and decides the settings for the current session in order to push them in a learning curve toward our desired goal of what we call a trained animal. And this is an example of the introduction of different levels of contrasts. So we train animals, we start with easy contrast and we introduce more complicated contrast as the time goes by, as a function of days of training. And we can get good performance of about 14 days, which is great, because if you take 2 years to train an animal, then it's not a viable task. And the color code is basically the same thing as the psychometrics. There's a proportion of right word choices. And so these are contrasts to the right, these are contrasts to the left. And this is what you want. You want the red on the left, and O on top, and blue on bottom. At some point, we realized that the 50% stimulus was not causing any difference from the 100% stimulus. So it was basically, if 100% is here, the 50% was here, so it was already asymptoted out. So we basically remove that, and that increases the number of, rather decreases the number of experimental conditions that we have, so decisions are more reliable. That whatever decision that we make will be more reliable. Okay, so then this is kind of the capture figure of the paper, which is this is the same. This is kind of mock data, and this is actual real data. These are all of the psychometric function of the average subjects divided, so different colors are lab. The take-home message from this plot is they all overlap, which is exactly what we want. So visually, I cannot see, okay, this is a Lisbon rat or mouse. Sorry, I still get rats confused because I did my PhD with rats and not mice. This is a Lisbon mouse. This is a New York mouse. This is a London mouse. We cannot do it by eye. So then we asked, we trained a bunch of, and we trained a lot of decoding models on the data to try and, okay, tell me, I'll train you with some data, I'll give you some test data, tell me what lab it came from. And so we have chance level here at I don't remember, I think it was 0.10 something for some other reasons we'll not get into. But when we feed the model the animal behavior, we can see that it's not different from chance. So all of the the decoding accuracy is at chance level. And we had, of course, to do a negative and a positive control. The negative control was a shuffle. That's what you usually do in these cases. You train the model with shuffle data. So it's kind of, you remove the structure that might exist in the data, and you retest the model, and you see that the results are very similar. And then we have to have a positive control, because it might just be the model that is not good enough for the data. So what we did is we added only the timestamp of the session starts. And so it's a UTC timestamp, so it has a time zone, and that is enough for the model to be able to decode where the data comes from. So this gave us some confidence that whatever behavior we were implementing in the animal, it was comparable across labs. And this was already very, very good because of the reproducibility crisis. So it is a reproducible task. We went through the process of, because we had to explain to other people how to build the rig, how to configure the rig, and how to train it. Well, training of the animal is mostly automated, but there's still a bunch of know-how that we have to share that is not necessarily in a computer. Surgery protocols is one of them. They also vary depending on subjects and on the people who training, which is also a problem for EFIS and for other more sensitive measurements of brain function. So, as I was saying, and reinforcing the openness of the thing, the paper is open access, of course. All of the code is in our organization on GitHub. We have a main data portal where you can look at all the data as it's coming in. And all the protocols are published on Figshare. Okay, so that's behavior, first step. And I don't want to take too long. The goal was always to record from the whole brain. So our recording plan at the beginning was, okay, let's record with neuropixel probes. So neuropixel probe is it's basically a shank with contact points that you put inside the brain in a chronic way, not a chronic way, sorry. In an acute way, so you have the animal head fixed, it's in the box on top of the wheel, and you insert the probe. We actually do two probes at the same time. It's a fairly new technology. The person who developed it is the only one in the world that I know that can do 8 at the same time. Well, first of all, we decided to do only half of the brain, so one hemisphere, which is something that I find very interesting in comparing humans with animals whenever we're doing neuroscience or neural stuff, is the lateralization of function is something not very well explored in animals. We just assume that the function are the same. So we don't have specific language centers well, I might be saying stupid things, but that I know of, language centers in, for example, mice, that you say, oh, Broca and Wernicke homologs are here, so if you remove just the left hemisphere, then the animal cannot speak, or there are some functions that we cannot see them do. Actually, rodents in general are very resistant to a bunch of manipulations. You can actually completely decorticate, these are old experiments done in the 60s. Then you decorticate rats completely, only leave everything that is not cortex, and let them recover. You put them on the floor and you compare them to other rats, you cannot see a difference. Now there is a question, what is cortex doing in rodents? That's a whole other discussion. So the idea was to have all of these insertion. There was some quantification of the expected data that we would get. It's a lot of data, so we're talking about 40 to 60 terabytes a year, and it's of course a moving target. We're recording not only behavior, we're recording, of course, electrophysiology. So we have 2 neuropixel probes at the same time, which is about 1,000 channels at 30 kilohertz. We're recording with 3 different cameras from both of the sides. I'll show you an example of tracking algorithms in a second. And one over top, so we can see the general movement of the animal. We have a ultrasonic microphone to look for vocalizations. We have a air temperature and pressure sensor also that measures every child. We can actually see when the experimenter opened the box because the pressure changes. And so we have a lot of data. That's the thing. And we have a lot of data that will probably go unexplored if we didn't share this data publicly. That's another motivation of actually sharing data publicly. So this is basically where we're at now. And so In terms of behavior, this is actually real data now. This is the same psychometric function with the shift in the psychometric that we see in different blocks. What we did have in terms of, so the goal is to do exactly the same that we did with the behavior, but for electrophysiology data. Electrophysiology data is much more sensitive and has a whole different view of issues that one must be careful. The stupidest of the ones is that normal electricity in the US is 60 hertz, in Europe is 50 hertz. So you have 60 hertz noise in recording from the US, but you have 50 hertz noise in recording from Europe. So these kind of things is stuff that you never think about. When you read the paper, you just look at spikes. There is a whole other effort, because there is a lot of us, there is a lot of available time to dedicate to these projects. It's like, okay, I never liked this, let's improve it. And there is a lot of kind of momentum right now, so that people will actually invest their time to try and solve global problems. One of the things we did, or we decided at the beginning, and we actually maintained, was to have at least, I think it was, let me just look, it was, okay, so the insertions in the brain have to overlap in at least two different labs for a specific number of sessions, so that we can actually compare and validate the data that you have with what is already known of that particular area. We chose visual areas because it's mainly a visual task. So we have about 78 probes in repeated sites, which is how we kind of standardize and homogenize all of the rest of the ePhys data. And then here is an example of, this is one trial. So here you have, you cannot see very well here, but here you have the moment of stimulus onset. Here is the moment of movement onset, and here is the feedback, either positive or negative, either a drop of water reward or white noise. And this is actual activity of the brain during these moments, and you can see that it changes. Well, you have to quantify this, but you can kind of see that when the stimulus comes up, the occipital areas kind of go up, and when the movement starts, you have more parietal areas that are responsible for movements. And we have all of the analysis that you would want to make on this whole data set, which is the hardest one to reproduce because it takes a lot of data, will be available soon so the data is already public, so you could just download the data and try it yourself if you want. We are in the process of publishing. This is actually in preparation. This is actually already posted in bioRxiv, if I'm not mistaken, and we're submitting it to Nature right now, which is the whole. You get a new ePhys paper. What are the analyses that you want to make? Oh, I want to see this. So we have an encoding and decoding figure. We have a manifold analysis figure to look at high-dimensional stuff and low-dimensional stuff. We have just normal quantification of firing rates and of spike shapes and of all of these things. So there is no real science in it. It's just really a good description of the whole data set, which I think is really required, especially when you do things this big. Otherwise, people don't know if they can use the data or not for whatever their purpose is. So besides that, here is this is an example of a whole of our video pipeline, which was a whole big effort. Because we have 3 cameras, we are actually reconstructing in 3D positions of the hands of the paws of the mouse, and we have a completely automated process. We are given a big image like this. It selects the relevant ROI, so the nose region and the mouth region, so we can get sniffing from the nostrils opening and closing, licking from the tracking of the tongue and the spout of the water, so we can see when the distance goes to 0. We have pupil, also dilation, and all of these are kind of time series, which we pre-process and give it for you. We tell you how we pre-process it, but you can download. You don't have to do it from scratch. Okay, so I take too long to explain things. This is a really short kind of review of our architecture, and these are kind of the things that I think we did well from the get-go, not because we're smart, I think we were very lucky. So one of the ideas that I think is very important is separate data from metadata. So we're collecting all these files and all the description of these files and how we describe these files. And instead of putting everything together in one place, which would make I want to download one session. We generate, 500 gigabytes a session of data. I want to just check the performance of the animal. I don't want to download all 500 gigabytes. I just want to download the relevant data sets. So we separated the data, and we have let's call it a public hard drive at the Flatiron Institute, which is an institute from the Simons Foundation, which is one of our funders, that generously gave us, I think it's 2.7 petabytes of space, and we basically dump all the data there. And we developed a database, but the database is only metadata. So the goal of this database is that it fits on a pen drive. So that I can just ask it questions, and it knows where all the relevant files are. So I can ask it questions like, give me how many mice do you have, first of all, how many subjects do you have, and how many male subjects do you have, because we try to have a balance between male and female mice. That's another thing in the literature which is weird. And give me all sessions that were done at 2 p.m. local time, and see if there is a difference with sessions that were done at 8 a.m. I don't know, you can ask these kind of questions, it will point you to the correct data, and then these are the things that we are developing and maintaining, which is we call it ONE, or ONE, is the Open Neurophysiology Environment, which is our API. So you ask questions programmatically, it downloads them immediately once you have the credentials for accessing the database. We have a mandate to release data either within 12 months of collection or upon publication, whichever comes first. And this is kind of important, so you don't actually sit on the data until you publish, and then you forget to release the data because you ended up already publishing. And so this mandate actually makes us have to have a system that is always working and doing several releases, also personal projects and such. And so it's pretty well tested, and it works pretty well. And as I was saying, the good thing of dividing data from metadata and having kind of this architecture is that there is no difference between internal and external user. We're developing the things that we are using every day, which makes the development go faster. But also, once you have to just open it up to the public, it's the same procedure that you already know and you use every day. So you don't have to change. It's not a whole process to, now we have to release and curate the data. The curation and the quality control is done as part of the pipeline already. Then we have tags and release and revisions, so we can play around with things. If we make mistakes, we can inform everyone. And the key here is not to make things, not have things wrong, is to be open with when you have it wrong and you figure out. So we dedicated a lot of effort in actually documenting all of this. So all of this is accessible for anyone who hopefully also is very kind of low code, or you don't have to understand AWS and web servers and all of that. Most of the things are explained, I think, in a pretty accessible way. We use Figshare for white papers, and we have a kind of a static release of the data, which is much more clunky, so it's not as agile to use, but it's kind of an archival format. We use Neurodata Without Border, I don't know if you're familiar with it, which is NWB. So we have converters that we're working with the developers of NWB, so to convert our data format in theirs. And we just dump it on Dandy with the current, and I believe Dandy has DOIs also, which is good. We have, these are a bunch of online resources for tutorials and courses. This is from our course we did in Cosine this year, actually. How to interact with our data and how to actually download and start asking your own questions to the data. And we ended up investing a lot of time, once you have a lot of data, in visualizers. And how you actually explore, now that you have this amount of data, how do you actually explore all these data? And so I'll go fast through this. This is more or less very contextual things that we're kind of trying to improve, like algorithm for clustering and spike sorting EFIS data, compressions, and this is submitted at the moment, I believe. It's called Lightning Pose. It's a way of, it's a semi-supervised pose estimation algorithm, or AI. If you put AI, it's easier to publish nowadays. But this came actually from how we're gathering the data from all of these cameras and using this automated pipeline. Now we have the problems of actually estimating clusters of positions, if you want to say. That's what pose estimation is. Here's a bunch of data. Tell me where to look at, because I cannot look at everything. So these are kind of an example of the tools that we're working on in terms of alignment of EFIS data. Once you collected the data, you want to know where exactly you were. We have a whole histology pipeline that would give a presentation in itself. How you actually plan your trajectories when you're inserting the thing, and it's a 3D model of the brain, and you actually set the position, and you can actually plan before having an animal in the rig. And a bunch of visualization tools and visualizers also of the raw data that comes from the devices. Anyways, okay, let's see if in 10 minutes I can talk about the social part. Okay, so this is what we're doing and what we started doing, and the motivation for what we did. Of course, this is a big collaboration. Again, it's about 70 researchers in 6 different countries. It's 8 to 10 different time zones, because it's +2 in Lithuania and -8 in LA. So it's 10 time zones. And it has advantages and disadvantages. So I want to talk a little bit about how we organize our research, how we organize our governance, and the infrastructure you kind of already saw. So in terms of research, the way we work is it's mostly a flat organization, and we divide stuff in topics of research. So we have working groups. Working groups usually have chairs and associate chairs. They have 2 chairs that are PIs, that are the ones leading the group, and 1 associate chair, which is basically a researcher, which is responsible for following up with stuff, creating agendas for the meetings, and such. And all of the groups, actually it's here, no. I don't know. Oh, yeah, it's here, it's the second line. All of the groups are open to everyone. So there is no, you see immediately all of the groups that exist. These groups appear, and some of them are more permanent, like the physiology working group is more permanent. The behavior working group was very active when we were publishing the behavior paper. Now it kind of only works if we have some problems with the behavior that we didn't notice until now. But these things are very dynamic. So they're created and they can disappear, and you can have ad hoc working groups. I don't know, me, you, and you, we have a specific problem. We create a working group, we schedule a meeting, and we start using a regular meeting to solve a particular problem. When the problem is solved, then we just go back to what we were doing. We have task forces, which I'll kind of talk about it in a second, and personal projects. And this is kind of all related. We use a bunch of kind of commercial software, mostly except for Zoom and Slack and Notion. Actually, these 3 are not open source. But we use these, and we use the Google Workspace, or Google, whatever it's called. It was Google Suites, now it's called Google Workspace, which is our main kind of infrastructure where we get emails and all of it. Slack turns out to be very important to give you a presence of stuff happening. There's always someone online because we have so many different time zones. And whatever time of day you can ask a question, someone usually answers, which is actually very cool. On the other hand, it's actually very invasive if you don't know how to actually set boundaries and configure it properly so it doesn't bother you in off hours. In terms of research, we had a big problem with credit assignments. So there's so many people in so many different countries, in so many different institutions. How do I make sure, 1, that we reach the goals that we want to meet, we want to reach, and 2, that people that are working with us, so students and postdocs, we treat everyone for the same, so they're all researchers, and there is not much difference between them. How do we make sure that they're better off by having participated in this collaboration than by not? Because our time scale is different, so the collaboration time scale is much different than a postdoc time scale. That sometimes, especially in the US, in 2 years they want to do the postdoc, want to move to a PI position somewhere, or to apply for other things. So one of the things that we kind of imposed is we have a publication working group that actually define the policies for how we publish stuff. Everything that is a collective effort, so we would call it a platform project, which is kind of described in the grant itself, and the main reason for why we exist. Authorship is very simple, is IBL is first author, everyone else in alphabetical order. The only difference is we have this concept of task forces, which are basically working group, but the task force is specifically designed to get a paper out there. So they're the people who kind of do all the, if the argument makes sense. Which figures, how many figures are we using? Are we submitting here, here, or here? Going back and forth with the editor. And these things take a lot of time and a lot of effort. And so people that usually are in task forces, the way we kind of motivate them to get to work for task forces is they get an asterisk when the paper is published as a corresponding author, which is kind of important if you're a corresponding author for a paper versus in 50 people. It's more important if you're a corresponding author, but we really wanted to get away from the first author, last author kind of dictatorship and single credit assignment thing. Besides that, well, I'm going to skip forward and backwards, sorry. Besides that, we're doing, this is called the credit taxonomy. The credit taxonomy is basically a standardized way of defining contributions to a particular publication. And so we kind of altered it because we are a particular publication and people are free to do it. These are all of the areas, and these are all of the authors of, I believe this is the behavior paper, and these are the contributions in terms of effort. The darker there is, it's 1, 2, or 3. I just tangentially did something here, I was equally contributing to this, or I was lead in doing either the conceptualization, methodology, there is a bunch of different things. You might argue that it creates more problems instead of less, but that's a different issue. The other thing that I think we did that works very well is because we are different time zones and we're distributed, we wanted to have kind of a zeitgeist of what was going on scientifically. Not only because we want to keep track of who's working on what, but also because we want to, your scientific identity is basically defined by what you're working on at the moment, right? So we wanted a way so people could say, oh, look, I'm working on this. So other people from the other side of the world could say, oh, I'm interested in this, let's collaborate. So we really wanted to incentivize collaboration between members of the consortium, students and postdocs, and also between theorists and experimentalists. And so the idea of this is the only thing that is private in the whole consortium is the project, and of course, HR stuff, but the project registration. And so the project registration is just a GitHub it's a GitHub issues thing. And the only thing that it does is you can dump three things there. Ideas, project proposals, and projects. An idea is basically, I read a paper, I'm working on this, I have this idea, this would be cool, someone did it. I put it out there, but I have no claim on it, and it's a declaration that I don't pretend to work actively on it. It's just something that someone should do. A project proposal is actually an elaboration of this idea, so it's something like, oh, I want to work on this, so it's a declaration to other people that you're actively thinking of working on this, and it's actually the first step on how we start collaborations, right? Because they say, oh, I want to do Bayesian analysis on how the brain encodes priors in this part of the brain. And say, oh, I know how to do Bayesian analysis well, and I know, let's do something together. And the project proposal starts to become structured. So it has a hypothesis, it has an operationalization, has an expected, it has authors. At some point, it will require a project board, which is other 2 PIs, possibly none of your local labs. And then it becomes a project. And the project is actually resources of the collaborations that are being worked on, that are being used, and particular ideas and hypotheses that have been tested. Okay, so in terms of governance, again, all of our policies are in this IBL governance. And so all of our policies in terms of data sharing, publication policy, how we deal with infractions or academic misconduct or everything that we can come up with are usually the product of working groups that they elaborate policies, and then there is a whole approval procedure. And the approval procedure is actually interesting because it's a consensus or a consent-based decision. It's not a voting, it's not a democracy. Basically, if we're all in a group together, and I have an idea, I present a proposal, the proposal is accepted by default. Unless someone has a specific objection that has to be addressed. What this means is that you don't penalize people for initializing something, and you say, okay, if you're thinking about this and you think this is important for us, yes, let's do it. That's the first answer, and this is at the kind of organization level, which I think is important. Unless someone says, no, this cannot be, because otherwise this is going to fail, this is going to fail, this is going to fail. So it goes back and it has to be addressed, and then it's presented again. But it looks like a slight change in terms of the decision-making process. It's not a voting part, so like we have the majority and the minority. We have people that are proactive and start doing stuff, that will start doing stuff and are allowed to do so. Unless there is something that someone sees that will actually impede something important. And finally, infrastructure. Yes, we have an amazing software development team, which is the kind of core for this, and there's a lot of the resources allocated here in terms not only of managing the software management team. We have a data science and a histology site researcher also that is only doing histology for all of our animals. We didn't reproduce histology because it's too expensive machines to do it. We do it in an automated way. And yeah, Times also talked about this. So this is kind of a call to come, please interact with us and use our stuff. The thing that we use, the more people use and the more it's useful, the more it gets developed and the more it's good for us because it's kind of it's our feedback. Besides of the paper that might come out, everything we centralize everything in our website, which is internationalmarinelab.com. Our tools and policies are here in our GitHub repository. And if you need interactions that is not Slack-based, because we have Slack but the rest of the world doesn't, there is this neuroscience.org, which if you just tag any questions about neuroscience with IBL tag, it will immediately be forwarded to our Slack and someone will answer. Usually there is someone, either staff or researcher, that will take the time to actually answer people from outside. This is very used when we do courses and workshops, because that's how people interact, especially if the workshop is a kind of hybrid format. And yeah, so now plans for the future. We have a lot of different modalities which are coming online right now. The idea is the same. Is okay, we can do this with behavior and electrophysiology. Let's look now at mesoscope and wide field imaging. Let's look at fiber photometry. Let's try to apply the same principles now that we have this kind of amazing infrastructure and amazing collaboration going on. Let's see if we can scale it up. Chronic neuropixel recording, some more genetic tools like BARSEC. But I believe fiber photometry has a whole neuromodulators project, which is having a view, same task, so same uninteresting task, but for the first time you can have ePhys and now neuromodulator activity in the same exact task, and whatever these, every technique has its limitations, you know that. And some things are good, some things are bad. But unless you do all of them in the same conditions, and you have the possibility of doing so, you will never actually troubleshoot or actually benchmark what these differences are. So this is what is right now in bioRxiv. I believe we're submitting it now, which is the ePhys counterpart of the behavior paper, which is, we call it GREAT, which is Guidelines for Reproducible Electrophysiology Across Teams. So where we're defining a set of indicators and quality control measures so that anyone can say, okay, I have a recording here, I have a recording here, can we put these together? Can these be comparable or not? And so wait for it, because it's coming soon. And I want to thank all of the team. This is a picture, I believe, of one of the first meetings that we had all together in Paris. I believe I am, here. I would like to thank, of course, the Wellcome Trust and Assignment Foundation with Flatiron Institute, specifically where all our data is, which are our main funders. Everyone at the IBL, and of course the Champalimaud, which is where all of this kind of started at the beginning. And I'm open for questions. Sorry, and maybe it took a little bit longer than I expected.
One silly question. You have a fairly well-developed system of preparing something like sonic. So you like authorship, contribution, posing a question, and all that. How did you emerge that? I mean, did you have somebody sitting down and say, well, this is the plan? No, obviously not. And it's quite interesting, because it's quite an intricate model you have here. And that doesn't come from one person sitting down and doing it. But the question is, how did the people collaborate on that? Because this is quite a complex issue, given particularly human motivations.
Yeah, and I think the key here was our kind of decision-making process. The idea that anyone who cares enough about any topic feels free to actually propose something is how you start then elaborating it. Then these things take a lot of, I don't know, we are at revision 6 of our publication policy and revision, I don't know how much of our data policy or of our...
How long did it take to start it?
To start it?
Mm-hmm.
I believe that the first... So the project was funded in 2017, so the year I finished my PhD. I started working in 2018, and in, I believe, February or March. And the first paper comes out in 2021. I think that kind of gives you an indication of how long it takes to ramp up, because there was a lot of problems to solve. And of course, there are asymmetric contributions at that moment that were probably not very well quantified. For example, I was basically in charge of the development of the behavioral task and of the experimental procedures. I did 80% of the work. Of course, there was a full working group that knew what they wanted, and it's kind of a feedback where, I did this, you have to check it, does it install here, does it install there, and all of these things. But that is okay, because I believe if things are clear from the get-go, this is what you're buying into. I think it was an experimental thing, so the whole, with some variability, but the whole way we recruited people was also, look, we're trying this new thing. It's not guaranteed that you'll have a great postdoc or a great PhD, but it's really cool and it's really interesting. Do you want to come on board? And so that actually gives you a lot of people that are very, not only highly motivated, but very engaged with the whole process. And in this case, the whole documentation of defining policies and discussing these things, if the people that care about are able to meet, decide things, and those things are actually how we do things by default, I think that gives enough. For me, it's super motivating, because today I want to organize how people should store files in their hard drive. I put a policy out there. If no one else has anything against it, that's how we do things. And from now on, we have about 70 researchers around the world, plus a bunch of labs that are using our system and a bunch of labs that are interacting with our way of doing things. Now it became a standard, right?
So you have no clashes?
No classes?
No egos?
No, we do. It's science. We have a lot of egos and very strong ones.
I mean, that's and the thing that it doesn't go well with the philosophy you have here.
Yes, I agree. I think that the reason why this is working. Okay, so a lot of the heavy lifting is done by the staff, and that turned out to be very important. Is to have people that are 100% dedicated to the project. Because PIs, yes, they have their own labs. This is one more project that they have in the lab. If it fails, it doesn't break their bank. But for people that are buying into it, that's their main investment, so they want it to make it work. But they're having people that are 100% responsible for understanding what's going on all around it, reporting back to the executive board and to the General Assembly, which is the meeting of PIs and the chairs of the working groups. That kind of helps you maintain some cohesion. And because all of the PIs, that they do have strong egos, but they usually are susceptible to rational arguments, which is one of the good things about science. Usually people are susceptible to rational arguments. But because they have less invested than everyone else in it, they also don't have very strong, it's not decision power. If you have API, which is actually pushing forward some particular way of doing things, that will become the way of doing things until you have another one that will push a different one. So things are not blocked. They don't have a way of blocking each other in a non-rational way. And that's it's something that comes from, I believe, the industry and corporation. It's called sociocracy 3.0. Apparently there's 2 before that. It's exactly this way of making decisions, which is a consensus-based decision. There is a whole way there's different types of objections that you can have to proposals and to policies, and that seems to work very well.
That seems to be like the Dutch government working. It's true.
On the other hand, I have to say, and how many people do we have on? Oh, this is being recorded. I'm not sure if we should say. But on the other hand, there is a little bit, if it were not for the excellent people that we have managed to hire all over, both staff as PhD as postdocs, I believe because none of the PIs is super invested, you don't have really a vision for the thing. The vision was this, which was not very, if you think about it, it's not a scientific project, it's a data-driven project. We're collecting data, we're doing something that is too hard for one lab to do, so we do it all together. The science is starting now. So we already have a bunch of spin-offs of people that are addressing specific questions, using our tools, using our this big data outside as kind of your proof of concept data. You say, oh, I see, and that's ideally what I would like is that in 2 or 3 years' time, I grab the ideal data set and go to first-year students of neuroscience and say, here is the brain in the same experimental conditions. Play. And so all of the questions that you might have that will not waste 2 years of a postdoc to answer, like what is the bed nucleus of the stria terminalis doing when the animal is not engaged in the task? I'm not going to waste a postdoc on that or a grant on that, but you can ask that question with this data set, just because we collected all of these data. So there is a lot of preliminary data and kind of proof of concept data which you use to ask to write actual grants to do your experiments that you can actually play around with our data sets.
So I mean, it's a very interesting organizational structure. But the other question I have, and it's basically highly sophisticated, reliable data collection.
Mm-hmm.
What is the most interesting scientific output you had? Did you have any?
So we have a lot of accessory, we call these kind of personal projects that appear. We have one on learning. For example, we discovered, well, one of standardization during the behavioral standardization phase, we played around with citric acids and water restriction stuff. And because we had a master student that was interested in that, she was actually training a bunch of animals. Oh, can I try? Of course you can. Can I try? So basically, it actually changed our water restriction protocol. Instead of restricting water, we actually give them free access to citric water during the weekend, and we give them sugar water in the. And there is a bunch of troubleshooting to figure out exactly how much acidic the water has to be, but they maintain a healthy level of water restriction that gives us enough motivation for them to perform the task and use this learning curve. That was one scientific output that came out of it, of the beginning phase. We have a bunch of them. We have, so if you go to our website, we have a list of publications, which are not the main platform ones, where you can actually see what the people that were contributing to these things were thinking, were trying to do, and how they use this infrastructure to test a particular hypothesis and so. Yes, it's a contentious issue. But this is not really, it's a data collection project. Yes, it is a data collection project, but I think that in the way of doing so, we open up not only, someone had to try it, because it's a data set that I really think should be out there at this point. I mean, we're in 2023 right now, and there is not EFIS resolution data set of the brain in the same experimental condition. Now there is. And I think that was my main motivator for joining the project at the beginning, knowing that it was not really specific scientific questions. But now I hope to actually leverage the time invested here to start asking interesting scientific questions. I did my PhD in spatial navigation using olfactory cues. It's hard, but I can look at orbitofrontal cortex and ask a bunch of questions on how space, not necessarily space, but how the visual cues are encoded, comparing it to so there is a lot of, it's like ChatGPT. If you don't know about a subject, you ask it, and it will give you a good idea. So if you don't know about how the brain works in a particular, how two particular brain regions work in a particular, in terms of information processing, you can ask our data sets. You will get an answer. It's probably not perfect, but it gives you an idea. And I think that's super important at the moment.
Thank you.
On that study was the mice. What does this design contains that other studies doesn't have that makes it international?
You have to repeat. Right?
The mice studies, what does it contain on the design that you proposed that makes it international reproducible than other studies didn't take into account?
The mice trained?
The mice studies.
Oh, the mice studies.
Presented, yeah.
It's reproducible because all of the data was collected simultaneously in different locations.
But the designs could be like any.
Yeah, but I had to describe it properly. We had to describe it properly. And the steps so that it would be reproduced. Usually what you get in reproducibility is I do something, I spend 2 or 3 years doing something, I publish a paper, and someone else grabs your paper and says, oh, this is interesting, I want to do the next step. And I want to redo it again, and then that's where the problems appear. What we did from the beginning is that we're all doing everything at the same time, so by design, everyone would have to do the same thing at the same time. So I'm very, very confident that if I give you my behavioral protocol and you buy the thing that you have to buy, we have a list of everything that you have to do, you can re-implement this task easily in a lab, in any lab. I mean, because we really describe all of the procedures from a first principle basis.
And other studies didn't do that?
Usually, no. You do your study, you publish your data, and then your rig might be used by some other student if you have some continuation going on in the lab, or mostly disassembled and used for parts in another setup for testing another hypothesis.